Kaye is a pharmaceutical chemist who has been very successful at translating novel ideas into profit-generating products. He established the annual Kaye Innovation Awards in 1993. The awards have earned an esteemed reputation, highlighting innovations with translational potential of income for the university, principally through royalties. Kaye joined the British Friends of Hebrew University and is a member of the Hebrew University Board of Governors.
My name is Yossi Paltiel. I am a professor of applied physics here at the Hebrew University of Jerusalem. And we are doing applied devices or doing quantum devices at the nanoscale, going very small, doing devices that will have quantum properties at room temperature. Every molecular in the nature is chiral, which means all life has some direction. It's like right hand and not left hand. All the proteins have one circular, are left-handed, all the sugars are right-handed, they are different. When we make sugars or protein or drugs or foods by chemistry, we have both sides. What we have found is a simple, generic way to separate between them, which means that if we use it in the food industry, the drug industry, the agrochemical industry, we can have better life, purer drugs, much less side effects in drugs and all of the food that we are consuming. My and my group's breakthrough is very important for physics, biology, and mainly chemistry, because we find a simple way to separate between the two chiral molecules, the two enantiomers, and this will be a breakthrough on improving drugs, agriculture, and food, because they will be purer and with much less side effects. So I want to thank the Kaye Foundation, and I want to thank everyone for joining me in this celebration.
Hi, my name is Gabriel Nussbaum. I'm at the Faculty of Dental Medicine, and my main interest is really understanding inflammation.
Hello, my name is Amnon Hoffman. My task is to take the drug from its intake into the site of action.
Hi, my name is Chaim Gilon. My main interest for the last 50 years is the conversion of peptides and active regions in proteins into drugs.
The advantage of taking a drug orally, a special peptide drug, is something very unique. It's very convenient clinically because eventually you'll have a drug where the patient can just have a pill, take it, swallow it, and it will have its anti-autoimmune disease treatment.
The breakthrough is the ability to convert an active region of this protein into a stable drug. No one's really been able to do that before. And this technology and the collaboration between our three groups really allowed us to make that breakthrough. We would like to acknowledge and say a big thank you to the hardworking students who contributed to this project. In particular, we'd like to mention Shira Dishon, and this was the main part of her PhD project. She's graduated, and in addition, we would like to thank Adi Schumacher in Amnon's lab, and Ala Talhami in the chemistry department. And of course, we would like to say a big thank you to the Kaye Foundation.
My name is Professor Oren Tirosh. I'm the director of the Redox Laboratory Lab in the Faculty of Agriculture and the Institute of Biochemistry, Food and Nutrition. And basically the lab has a main focus on two types of research. One is in nutritional sciences, focusing on fatty liver disease, and the other topic that the lab is focusing on is in food science, basically on food preservation of meat, and this is more or less the topic of our new kind of technology that we developed here that is supposed to give us a better and safer preservative for meat and for processed food, which will make, I think, a revolution of our food health products that we can get from the industry. Eventually, our vision is to form kind of a processed meat that when we are adding the compound, it will be even healthier compared to fresh meat. It's important to mention that the group that is working on the project is the PhD student Adi Speiser, which is now almost at the end of her PhD, and my colleague, Professor Yossi Kanner, who is helping me with the development of this new technology.
Hello, I'm Josh Moss, an MD PhD student at the Hebrew University. I perform my research in the laboratories of Yuval Dor at the Faculty of Medicine and Professor Tommy Kaplan at the School of Computer Sciences. We developed a new kind of blood test analyzing DNA molecules circulating in the blood, which inform us about tissue-specific cell death going on in the body. By detecting elevated levels of DNA molecules in the blood from a specific location in the body, this may indicate a disease process going on at that site. We found that you can use this test to detect and monitor diseases such as cancer, heart disease, and diabetes. We believe that this blood test has the potential really to change the way we practice medicine and hopefully help many patients who are suffering throughout the world.
Hi, I'm Betel Cohen-Wiesel, and I'm from Professor Liaz Etgar's lab in the Institute of Chemistry in the Hebrew University of Jerusalem. In this current research, we combine together two-dimensional and three-dimensional structures of perovskite, which gave us better stability of this kind of solar cells, higher performance, and high voltage. And this is very unique for the solar cells, which till now gave us only 0.7 volt and was very limited in their applications. The breakthrough in this research is combining together two-dimensional and three-dimensional structures of this perovskite, which enable us to use the high stability of the two-dimensional structures, combining together with the high efficiency and better electronic properties of the three-dimensional perovskite structure.
Our university is deeply in debt to both Isaac and Myrna for their deep involvement and devotion to this institution.